Next up we have Ziminski, who is going to be talking about food, which is a little unfortunate because everyone in here is hungry. A postdoc in, let me see if I can remember how to say it, Koya's lab, part of the behavioral neuroscience unit in the School of Psychology, examines how the brain encodes reward-associated cues, and particularly for food, cravings for food. Okay, and you are leaving after being here for 10 years, so you do undergraduate here.
It's not very fun.
But leaving graduate after 10 years is a whole new experience.
Yeah.
But you don't know where you're from.
No, yeah. Cool,
no more comparison, right? And the title of Joe's talk is Hungry, Understanding the Brain's Response to Food-Associated Cues.
So as we navigate the world, we come into contact with thousands of different stimuli every day. So different objects and different contexts. And the majority of these stimuli are neutral, kind of psychologically and emotionally neutral. You don't particularly feel one way or the other about them. For example, I doubt you're particularly scared, say, of the projector when you came in, or excited by the chair that you sat down in. But there are some stimuli which can elicit such reactions, and we call these environmental cues. And I'm going to talk, as you know, about one kind of cue that we're all very familiar with, and these are food-associated cues. So food-associated cues can be any kind of stimuli that you associate with a food that you really like, and these are typically high-fat or high-calorie foods. So these may be the packaging of your favourite crisps or sweets, or the logos of your favourite high-street food chains. And these cues are particularly interesting because they can, well, they're attention-grabbing, and they can elicit cravings for the food that the cues associated with. They can elicit general cravings for food, and they engage our motivational and even our motor systems to make us want to approach these foods and consume these foods. So this may be why, say it's the end of the day, you've just eaten dinner and had pudding, and you're feeling fairly full when you sit down in front of the telly, but then an advert for your favourite crisp or sweet comes on, and all of a sudden you're not quite as hungry as you thought you were, and you're up out of your chair maybe checking the cupboards of the kitchen. And so it's no surprise that these cues play an important role in overeating and in obesity. And we know that people who have obesity show a hypersensitivity to these food cues. They report higher cravings and they show a higher brain activation. So what we're interested in Esika's lab is how the brain encodes these food-associated cues, and whether it does it differently to how it encodes neutral cues. So we know that food-associated cues are at least partly encoded in a brain area called the nucleus accumbens. So if you take someone and put them in an fMRI brain scanner and show them images of food-associated cues, you typically see an activation of this brain area. But interestingly, animal studies suggest that it's not the whole nucleus accumbens which is becoming activated, but only a subset of the neurons within it, typically around 5%. So it's kind of surprising that although these cues can exert such a strong control over behaviour, they're actually only activating a fairly small proportion of the total number of neurons in the brain. And we can access this experimentally by using transgenic mouse models whose neurons light up a bright green when they become strongly activated. So there, well, the green ones are strongly activated neurons, and the red ones are surrounding or less activated neurons. And so what we can do is take food-associated cues or neutral cues, show them to our animals, and then we have a situation where we can investigate these special sets of activated cells. So that's what we do. We have an auditory cue, it's just a clicker, that is neutral at the beginning of the experiment, and we associate this with a 10% sucrose sugar solution, which these animals really like. And then at a later time we can play this now food-associated cue back to the animals, and they go running to the position in the boxes where they previously got food. In another group of animals, we don't associate this clicker with any sucrose, and so it remains neutral. And now, because they're these special transgenic mice, we have a situation where in half our animals, the neurons activated are activated by a food-associated cue, and the other half of the animals, the neurons activated are activated by a neutral cue. And then we can go on to do our primary measure, which is electrophysiology. This is a way of measuring the electrical properties of these neurons. And you may be familiar with electrophysiology because it's what a cardiologist would use to measure heart rate in a hospital, so you may be or in a hospital. And you get these typical action potential waveforms from the heart, which you may recognize. We see exactly the same thing, but instead of measuring the heart, we're now looking at these select neurons in the brain. And so what we found was that the neurons activated by a food-associated cue, firstly, there was slightly more of them, and secondly, they were much more electrically excitable than the surrounding neurons, and the neurons are activated by a neutral stimulus. So this means that they're more likely to fire, and when they do start firing, they fire much more strongly, and so they're more effectively able to communicate with the brain areas that they're connected to. And this is particularly relevant in the nucleus accumbens, which is where these studies were done, as the nucleus accumbens is interconnected with many brain regions involved in emotion and motivation and even motor planning. And so what we think this is kind of like doing is, if you could imagine what the nucleus accumbens is a little bit like, it's like a giant classroom with hundreds of thousands of school children in, all trying to get the attention of a teacher sitting at the front who represents the brain regions the accumbens is connected to. And what increasing the excitability is a little like doing is making the 5% of those children who are trying to get the attention of the teacher to talk about the food-associated cue, is making them much, much louder. So the teacher's more likely to hear these children over the background noise. And so what we'd next like to do is, using some new technologies, try and reduce the excitability just of these neurons and see if we can reduce the effects of the cue over behaviour. And eventually, this is a long way in the future, but it would be great. As I previously said, people with obesity show hypersensitivity to these food cues. If we could find a way to target these neurons and reduce the excitability, maybe reduce the cravings that they experience, which are so fundamental to overeating. Thanks.
Quite a bit of time for questions.
This is a really difficult question based on your last statement. How on earth would you do that in humans?
Well, yeah, that's why I said it was a long way in the future, but I guess it would be like a combination of viral vector delivery of some kind of, it wouldn't be an ion channel, but something you could reduce the excitability of the activated neurons by a viral vector, which might be like molecularly caged, so that you could uncage that viral vector just in the nucleus accumbens. I mean, the technology is kind of there, but obviously in humans it's like 50 years in the future. But it's a nice idea.
So would you expect to see different levels of excitability depending on the cue, whether it was positive or the motivational? Yeah. So if it was positive or negative, if it was cocaine versus food or sex, would you expect to see changes in this, or do you think this is a reflection of something's good and this is a threshold they get to?
Well, we see similar things with drugs, with cocaine, in a very similar experiment in a slightly different brain area. So this brain area, the excitability wasn't increased, but a different brain area was. The neurons were. So it's difficult to tell, but it does seem to be replicated in not just our lab, other labs seem to be seeing this in a few areas. So it might be a mechanism by which these kind of salient stimuli can kind of capture or increase their signal to become more salient or to stronger control over behaviour. We've not done anything with if the valence was negative, with like fear. And in the accumbens, there are some neurons, dopamine neurons, which reduce their firing to a negative valence. So you might expect to see something where the excitability is decreased, but it's difficult because how we do it is based on activation. There's currently no way of tagging a neuron that's becoming less activated. So it's a question for the future, but yeah.
What are the different types of neurons in the nucleus accumbens? And are the cells that are becoming more active and more excitable all of the same kind?
Yeah. So there's three main types of cells in the accumbens. The majority are medium spiny neurons, which are GABAergic neurons, and they project outside of the accumbens. So about 95% are medium spinies, and this is all done on medium spinies. There are some GABA interneurons and some acetylcholine interneurons, but we didn't look at those. So yeah, this is all the same kind of neuron.
Just giving him exercise. So you used a noise, which is a bit like a dinner bell, saying it's kind of like time to eat, so an anticipatory cue. Do you get the same things if you use a visual cue, which is more like something you might eat, a consummatory cue?
Yeah, that's a good question. We don't know. What we did with drugs was a contextual cue, and we saw a similar thing in the nucleus accumbens for the core. So it doesn't seem like it's restricted just to these kind of signalling auditory cues, but we've not done anything with a light cue, and I'm not sure that I know about anyone that has, but that's an interesting suggestion.
Thank you very much.
Cool, cheers.